Hello, and welcome to another tutorial for BIOS E1A. In this tutorial, we'll work through a practice problem about 2,4-dinitrophenol, also known as DNP. As we learned in the course lecture, DNP is an uncoupling agent. While walking through this problem, we'll revisit how in the mitochondria the processes of aerobic respiration and electron transport are coupled to energy capture via ATP synthesis. DNP disrupts this coupling, hence its title of uncoupling agent. But before we get into the specific function of DNP and tackle parts A, B, and C of this problem, let's first look back and quickly review what we learned in lecture about the electron transport chain and about chemiosmosis and ATP synthesis in mitochondria. These processes will take place in and across the inner mitochondrial membrane, which separates the mitochondrial matrix from the intermembrane space. Here, in cartoon format, are the protein complexes that make up the electron transport chain. Electrons are delivered to the electron transport chain by the electron carriers NADH and FADH2, which were made when NAD+ and FAD were reduced in earlier steps in the catabolism of cellular fuels like glucose, for example in the TCA cycle. NADH gives up electrons to complex 1 and is oxidized back to NAD+. FADH2 gives up electrons instead to complex 2. Energy released from electron transport down the electron transport chain powers the pumping of protons, H+, across the inner mitochondrial membrane from the mitochondrial matrix to the intermembrane space against its concentration gradient. Only electrons from NADH pass through complex 1 and power proton pumping from that complex. But as electrons move through complex Q or ubiquinone and on down the chain, electrons delivered by both NADH and FADH2 power proton pumping from complex 3 and from complex 4. At the end of the chain is where oxygen is finally consumed. Electrons that pass down the electron transport chain reduce oxygen and, along with some protons, form water. Pumping protons from the already more basic lower concentration of H+ matrix to the intermembrane space establishes an electrical and chemical gradient across the inner membrane, which also gets referred to as the proton motive force. It is this proton electrical chemical gradient that couples the previous processes of cellular respiration down to the end of the electron transport chain with capture of energy via synthesis of the cellular energy currency, ATP. The electrochemical gradient set up by the electron transport chain drives ATP synthesis through an enzyme and molecular motor, aptly named ATP synthase. Diffusion of protons down their electrochemical gradient through ATP synthase powers its synthesis of ATP. Now enter DNP. How does DNP disrupt the coupling of respiration to ATP synthesis? As you may recall from lecture, DNP is an ionophore that can transport protons across the inner membrane back into the mitochondrial matrix. Given what we know about how crucial the H+ gradient is to coupling respiration and ATP production, now we can start answering the practice problem. I'll work in reverse from part C backwards. How do we explain DNP's uncoupling effect? Well, we explain it by the fact that because DNP moves protons across the inner mitochondrial membrane, DNP can dissipate some of the proton gradient established by the electron transport chain, the same gradient that normally powers ATP synthesis. In other words, the proton electrochemical gradient couples respiration to ATP production, and DNP short-circuits that process, discharging the electrochemical gradient and uncoupling them. Part B then asks, when it's doing its thing, ferrying protons across the inner mitochondrial membrane, why does DNP lead to an increase in temperature and to profuse sweating? We can answer this, too, with what we discussed about the electrochemical gradient. The electron transport chain transforms energy captured from catabolism of glucose into an electrochemical gradient in the mitochondria. This energy is then usually captured via ATP synthesis, as protons diffuse down their gradient through ATP synthase. DNP bypasses this. However, the energy from the proton diffusing down its gradient still needs to be released somehow. That somehow is as heat. So as a result, DNP transport of protons across the inner mitochondrial membrane down the gradient releases energy as heat, increasing body temperature, which then subsequently explains the profuse sweating response. Finally, Part A wonders, why would DNP cause an increase in metabolism? That is, increased oxygen consumption and increased catabolism of foodstuffs, also known as energy-rich molecules like glucose. As we've worked through, normally the electrochemical gradient drives ATP synthesis. DNP discharges some of the proton gradient. The result is a reduced generation of ATP. Metabolism increases to try to increase or restore the proton electrochemical gradient and increase ATP production back to normal. How do you ramp up the creation of the proton gradient? You run more electrons through the electron transport chain. That entails the production of more reduced electron carriers, NADH and FADH2. That also results in increased oxygen consumption at the end of the electron transport chain. Increased catabolism of food molecules and increased rates of glycolysis and the TCA cycle would also happen upstream of all this to generate the increased electron delivery to the electron transport chain. That concludes this tutorial for BIOS E1A. As always, a reminder that if you have additional questions, you can post them to the course discussion forums online or bring them to evening TF discussion sections. Thank you very much again for listening.